Hello students, in our previous class we were discussing about the metabolism, turnover, metabolic pathway, catalytic reactions, metabolic pathway. Yes, there are different words were there. So the same time we also discussed about the metabolic basis for living, nothing but anabolic reactions. So that anabolic reactions which include two things. One is metabolic pathway. In that there are two things. One is anabolic pathway, another one is catabolic pathway. Refers to anabolic reactions, and one more is catabolic reactions. So both together called as biosynthetic pathway. Yes or no? And we also discussed about the equilibrium state of body constituents. So that in today's class we are moving on to the one more very important topic, that is enzymes. So now look at me, what mean by enzymes, how they are, where they are. So these all things we are going to discuss in today's class. First of all, enzymes refers to, these are the biocatalysts. First and foremost word is, these are the biocatalysts. Here the word biology or bio refers to, they play a very wonderful role in the reactions, and second one is catalyst refers to, they can increase the rate of reaction. Simple. So the catalyst refers to the something which can enhance, which helps in for the rate of reactions. So that here the word biocatalyst means those catalysts can enhance the rate of biological reactions, are considered as biocatalyst. So, and one more very important word is that whatever the enzymes you take, whatever the enzyme or enzymes you take, those all are considered as proteins. Please do remember, I mean to say that single sentence is that all enzymes are proteins, but not all proteins are enzymes. Once again, all enzymes are proteins, but not all proteins are enzymes. But here the statement is, all enzymes are exclusively protein, except there are two things. One considered as ribozymes. Please do remember, important word, ribozymes. Ribozymes means these are the nucleic acid breaking. These are the nucleic acid breaking or digesting enzymes. The thing is, no doubt these enzymes acts as, these ribozymes acts as enzymes, but they are not made up of proteins. Do remember. So remaining these two, it may be DNAs and one more is RNAs. Usually the name of the enzymes ends with ASE. As understood. That is, say for example, you take lactase, yes or no, and one more is sucrase. Then many things are there. So protease, then carbohydratase. So most of the enzymes ends with the ASE. Likewise, here ribozymes refers to DNAs and one more is RNAs. Those are, act as enzymes, but they are not made up of proteins. This is one thing. And one more. See, as I told you right now, the enzymes are made up of proteins. In that, during protein synthesis, simple sentence, during protein synthesis, what happens? Soon after the protein synthesis, it will be a long light chain, as you know that. Already we discussed in that. That is a primary structure of protein, secondary structure of protein, tertiary, quaternary. So many are there. In that, imagine there is a long chain of amino acids. This is first amino acid, second, third, and so on. And these all amino acids are linked by peptide bond. Now this is considered as primary structure of protein. Simply considered as primary structure of proteins. See, all enzymes are proteins means enzymes also made up of amino acids. A simple thing. In that, this primary structure of protein never perform any biological function in the body. Do remember. Yes, once again, these, the primary structure of protein or the enzyme cannot perform any function in the body. So that now we'll take one more, a long chain of amino acid, and one more there is a long chain of amino acid, nothing but A chain and this is B chain. In that, these two chains are held together by, as we know, that disulfide bonds. These two chains, that is A chain, I mean to say that in this, A chain is nothing but a polypeptide chain of a protein, and B chain is also made up of amino acids, again one more chain. So that these two different chains are held together by disulfide bonds. Now this can make the secondary structure of protein. It can make the secondary structure of protein. Same time, the same second structure can be, this is like this, again A chain and this B chain, yes or no? And this one, and also the second structure can be helically arranged. I told you many times. Yes, like this. So here there is a binding of the same amino acids. So nothing but which makes, there is tertiary structure of protein. This is tertiary structure of protein. See, as I told you right now, the primary structure of protein never perform function as well as this one. However, the secondary or the tertiary and one more is there, that is quaternary structure of protein can be made functional. Yes or no? So this is our concept. In that, what I am trying to tell you is that during protein synthesis or during enzyme synthesis, whatever you take, first it is going to prepare the long chain of amino acids. Nothing but always primary structure of protein. Yes or no? So during, this is during translation or during protein synthesis. During protein synthesis, always it is going to form in the form of primary structure only, but it never perform any work. So for that reason, after protein synthesis, this whatever the protein is prepared right now, that undergo some structural modifications. Please do remember the word. It undergo some structural modifications. So what kind of structural modification? Like this. Can you understand this point? So the same amino acid, nothing but imagine it is made up of 100 amino acids. Just imagine it is made up of 100 amino acids. In that, the same structure of amino acid can be like this. Yes or no? So here, first amino acid, fifth amino acid. The fifth amino acid can be bind with the, however, 20th amino acid, again by means of disulfide bonds. Yes or no? Again fifth means here you can imagine 15th, again it can be 30 and last 100. So these can be bind each other. So for that reason, it can coil itself. So like you can tell like the woolen bulb. Yes or no? Woolen bulb. So that it can, now it can be become tertiary structure. Now it can be perform any biological function. See, this is one thing. At the same time, we have very important concept, that is active site. Very important word, active site. So the what mean by active site? In relation to this enzyme, what mean by active site? Very simple to understand. See, when during protein synthesis, as I told you right now, there must be a long chain of amino acid called as primary structure of protein. So during some structural modification, because it has to undergo some structural modification to perform function. So during structural modification, this long chain of protein can undergo structural modifications like this. Imagine. Yes. So here it look like a pocket. Yes or no? It look like a pocket or the crevices. So that during structural modification, within the enzyme or the protein, some pockets or the surfaces are formed. So that pocket is called as active site. Do remember the word. It is called as active site. So in that active site, the substrate is going to bind. Yes, the substrate is going to bind. Yes. Now the final conclusion is that what is active site? In the enzyme where the active site binds is called as active site. See, simple example I can give you. Imagine it is enzyme. Whatever the enzyme you take, this is considered as enzyme. See, during metabolic reactions or during enzymatic reactions, I mean those chemical reactions can take place in presence of enzyme called as enzymatic reactions. So during enzymatic reaction, this is, imagine, enzyme E, and imagine the whole enzyme is nothing but, sorry, the whole structure is nothing but enzyme. In that, particularly at this position is considered as active site. Yes, in the whole enzyme, why I am going to call it as active site? Why particularly at this area is considered as active site? Because when this enzyme binds to a substrate, clear this? So, and this is nothing but substrate. So this particular substrate is going to bind at active site of the enzyme. So for that reason, this active site is meant for to hold substrate. Hence, within the enzyme, see, as I told you right now, the whole structure is nothing but enzyme, but very specifically the substrate always binds to active site of enzyme. Then only it can break or it can form the bonds. So this active site, that active site is formed in the within the what enzyme. This is it is like we have many concepts based on this one, that is lock and key model. Lock and key model, nothing but a particular lock is opened by the particular key. So that this is what sometime we will be having like this. Imagine it is enzyme, same time this is what substrate, yes or no? So the substrate can be particularly fit within the enzyme. So nothing but this particular part is nothing but active site. Hope you understand this. So this is the enzyme. At the same time, the continued part is we understood the substrate and enzyme. So always substrate binds to enzyme at its active site. See, this is, imagine the whole hand is nothing but enzyme. The whole hand is nothing but enzyme. Particularly imagine it is a substrate. This is enzyme and this is substrate. When I put on this hand, it is not possible to fit here, yes or no? So for that reason, particularly I am going to put at this position, nothing but this is nothing but active site. Do remember? In this, when I take enzymes, there are two types. When I take enzymes, there are two types. One is enzyme catalyst. When I take catalyst, enzyme catalyst. One more is inorganic catalyst. There are two things. One is enzyme catalyst, another one is inorganic catalyst. So the very fundamental difference between these two is, again, just a minute. See, the fundamental difference is that when we take the enzyme catalyst, so they are very sensitive to what? Temperature and pressure, as you know that. Yes. So they are very sensitive to temperature as well as pressure. I mean to say that they can make functional, they are functional below 40 degrees Celsius. So once the the temperature of the body or temperature reaction cross beyond 40, they become inactive or they become denaturation. So this is, I mean to say, these enzyme catalysts can be functional only at the narrow range of the temperature or the pressure or the whatever the environmental condition. So they cannot perform any function beyond the particular the range. Yes, this is about enzyme catalyst. But thing is, some of the enzymes, no doubt enzymes only, but some of the enzymes can be isolated from some of the microbes which are normally present in the extreme environmental conditions. Please do remember the point. What? These enzyme catalysts can functional below 40 degrees Celsius. 40 is considered as optimum. Next, see, the same time what I am trying to tell you is that some of the enzymes can be extracted or isolated from the microorganisms, and those microorganisms normally present in the or normally existing in the extreme environmental condition. Nothing but hot springs. Example, hot springs, as we know that, and second one is thermal vents. Hot springs. Second one is thermal vents. Better word is methanogens, as we know that, methanogens bacteria. So there the same enzyme can be perform the function almost 80 to 90 degrees Celsius. They are still functional till 80 to 90 degrees Celsius. This is about what? Enzyme catalyst. And some of the enzymes or the catalysts are there. They are simply called as organic catalyst. Here they are not sensitive. Here they are sensitive and they are not sensitive. Not sensitive to temperature and pressure. No, they are not what? Sensitive. I mean to say that still they can perform any kind of function even though it is 80 to 90 degrees Celsius. Even though it is 80 to 90 degrees Celsius. So this is what the capacity of inorganic catalysts. Clear? So this is about one of the point regarding the catalyst. So when they ask catalyst, this is enzyme catalyst, another one is inorganic catalyst. Very simple is the enzyme catalyst can perform the function in narrow condition, they can be in broad condition. Same time, some of the enzymes catalysts can be extracted from the microorganisms. Normally they are present in the extreme environment. Still, even though it is an enzyme, but it can perform the function up to 80 to 90 degrees Celsius. This is what? The enzymes. I hope you understand the concept. Very simple to understand. Yes, we have one more topic, that is nature of enzyme action. Yes, nature of enzyme action. I mean to say that what kind of reactions are there? What kind of reactions are there? So nothing but nature of enzyme action. See, whatever the enzyme you take, it may be inorganic or it may be enzyme catalyst, whatever you take irrespectively. So how they act on the substrate? The very simple fundamentally we need to discuss. When we have a substrate, that substrate when binds to enzyme, definitely the enzyme can convert the substrate into product. Standard formula. Do remember. Yes, when we have a substrate, I mean whatever the material which is converting into product is called a substrate. Whatever the material which is converting into product is called as substrate. That substrate binds to enzyme and finally converted into product. This is our standard formula. So based on this one, so till end of this topic we need this one. Next, see how many types of enzyme natures are there. See, first one we have we'll discuss simultaneously. We'll discuss simultaneously. So first one we have physical change. First one is physical change. See, here the word physical change means, I mean what kind of enzyme action it is in that. See, first one is physical change means some of the material, nothing but substrate. See, so whenever I take the word material means, so that is converting into product is considered as substrate. So that substrate is being converted into product, yes or no? So that is influenced by the enzyme. See, in that the mode of action or the nature of enzyme action is physical change. In the physical change, what is going to happen? When we have a substrate, that substrate is remain as a substrate, no doubt. See, no doubt it is an enzyme action or the enzyme function, but thing is, in the physical change, substrate as a remain substrate. But the very important thing to understand is, when we have a substrate in the shape, that is like this, imagine the square, yes or no? So after the enzyme action, so it become irregular. This is what? Change in the structure or change in the structure of the substrate from one structure to another structure without breaking the bonds. No breakage of bonds. I hope you understand this. Is here the what is thing is going to taking place? Very simple. So initially the substrate is square. In presence of enzyme, the square become irregular, square become round, square become oval, square become rectangular, whatever the position or whatever the structure. But thing is, the square become, I mean to say, the one structure of the substrate is being converted into another structure of the substrate without breaking the bonds. Here there is no breakage of bonds, just it is going to change its shape. Hope you understand. This is one type of chemical reaction. Second one, physical change. Second one is change in state of matter. Yes, second one is change in state of matter. See what is going to take place here. Very simple to understand is, again here we have a substrate. Yes, change in state of matter. So without the help of enzyme, just substrate is being converted into substrate only. Substrate is being converted into substrate only. But thing is, this substrate was present in the one state after completion of the chemical reaction. After completion of the chemical reaction, it is converted into another state. See, better example I can give you. Say for example, we have water. Shall I write liquid state? Definitely liquid state. So when you keep it in the deep freezer, when you keep this water in a deep freezer, no doubt the liquid state of water is being converted into ice. Yes or no? It will become solid. It will become solid. See what is going to take place here? Same substrate, that is water, is being converted into ice, from liquid to solid. When you heat the ice, it will become vapor. Yes or no? Become gas. Yes, so no doubt here and here and here, in all cases we are going to observe water, but thing is, only there is a change in the state of matter. This is what one type of the reaction is taking place in case of any chemical compound. Okay? And this is enough for today's class, and remaining things we'll discuss in tomorrow's class. Thank you very much.